My postdoc Andy tried a simple, straightforward experiment to make the major vault protein in a cell. This accomplishment would be like getting a single in baseball. He failed miserably. Instead, to our surprise, he hit a home run. He made vaults. Hi, I'm Lenny Rome, the Vault Guy. I'm a cell biologist and a nanotechnologist. My passion is a common particle of the human cell called a vault. In our last lesson, we introduced Cryo-EM and how we use this technique to solve the 3D structure of the vault at low resolution. We also mapped the location of the various vault components inside the vault. If you haven't seen it, or if you don't know what an angstrom is, I'll put a link below. Today, I want to cover three things. First, I want to tell you about protein expression, a method that uses recombinant DNA technology. Second, I want to explain how we use this method to make empty vaults. Finally, I want to show you how we determine the location of the major vault protein in the vault. Stick around. I might have more pictures going up on the wall today. Around 1997, Dr. Andy Stephen joined my lab as a postdoctoral fellow. Andy received his PhD in biochemistry from the University of Liverpool, and he is currently a group leader at the National Laboratory for Cancer Research in Frederick, Maryland. Andy wanted to study the major vault protein, MVP, so he set out to make pure MVP protein. In episode 7, I described how to make a protein by translating its gene sequence. To understand what Andy did, let's take a simplified look at protein expression. If you have a chunk of DNA, and that DNA is the gene for a particular protein, let's call it protein X. You can slip that DNA into a cell and get that cell to transcribe that DNA into mRNA and translate the mRNA into a protein. Protein X in this example. That is protein expression. Andy wanted to make MVP in a cell that didn't have vaults, so he first tried the common lab bacteria E. coli. Recall, bacteria don't have vaults. Andy carried out basic bacterial genetics to insert the MVP DNA into E. coli, and he looked for expression of the MVP protein. To his dismay, he couldn't find any free MVP protein produced. We worried at the time that the protein might not be stable in bacteria, and that perhaps we would need to use a eukaryotic cell. One protein expression system that Andy was familiar with is called the baculovirus protein expression system. This system uses insect cells. These are eukaryotic cells which also did not have vaults, so he tried that system as well. What Andy found was very interesting. Andy inserted the MVP gene into an infectious piece of DNA called a bacmid. When the bacmid carrying the MVP DNA was added to cells grown in the lab, the cells didn't make any free MVP protein. But to our surprise, they made lots of particles that looked exactly like vaults. We knew that MVP was likely an important contributor to the structure of the vault. However, we were shocked to see that vault-like structures formed entirely from MVP. Before this experiment, we guessed that MVP was just the barrel staves. Now we saw that MVP could form the whole barrel. We published Andy's data in the Journal of Biological Chemistry, among the highest-ranked journals for biochemistry research in the world. We were delighted that the paper was featured on the journal cover. So let's place it on the wall with the others. I guess I'll have to find another spot for my painting of the Reichstag dome. Andy isolated the vaults that he made in insect cells, and he showed that they were entirely formed from multiple copies of MVP. To see how close their structure was to native vaults, we turned again to Cryo-EM and our collaborator Phoebe Stewart. Phoebe convinced two of her new doctoral students, Yeshi and Miriam, to pick up the Vault Cryo-EM project and to generate a structure for the vault that Andy made in insect cells. The structure of this MVP-only vault was nearly identical to the cryostructure of native vaults that we had previously solved. However, there was a difference, and that was that there was much less density, or stuff, inside. This supported the idea that the entire vault outer shell was made from multiple copies of MVP, and that the other vault components, TEP1, VPARP, and the vault RNA, were passengers bound tightly inside. Our next challenge was to figure out how the MVP protein was oriented in the vault. To do this, we made a vault particle in insect cells that was assembled from an MVP sequence that had been engineered to have extra amino acids attached to one end of the protein. Let's assume that this long red squiggly line represents the MVP protein viewed from a distance. If we could zoom in, we would see the sequence of amino acids that are attached together like beads on a string. Here we see the first 7 amino acids. The beginning of a protein is called the N-terminus. If we magnify the last 7 amino acids, this end is referred to as the C-terminus. We started by adding 31 extra amino acids to the N-terminus, illustrated with this pink squiggle. When Yeshi and Miriam generated a cryostructure, the outside of the vault looked exactly like it had when there was no tag added to the MVP. The red vault in this illustration. We were able to locate the position of the tag by carrying out a difference map. Essentially, the computer subtracts the vault with no tag from the vault with a tag. The difference density revealed that the extra density, the tag, was at the waist of the vault on the inside. A cut version, where the dotted line is turned on end, shows the center of the vault, and that pink area is the difference density. In other words, the location of the tag. We also made a vault from an MVP that had 12 amino acids added to the C-terminal end, illustrated by the green squiggle. A cryo-EM structure of this vault showed 2 blobs of extra density, one on each end of the vault. This allowed us to explain all of our difference mapping and tag data. We produced a model that proposed that the MVP aligned along the outside of the vault, running from the waist up to the cap. We published this data in 2004 in the Journal of Molecular Biology with Yeshi and Miriam as co-first authors. Once again, a vault image was selected for the journal cover. Let's get this on the wall too. All of these cryo-EM reconstructions I've been showing you were carried out on thousands of images, giving good resolution, but still relatively low, in the neighborhood of 20 angstroms. Although cryo-EM was getting better, if we wanted to be able to see the individual protein chains of the vault, we would need to go to a higher resolution. So we turned to X-ray crystallography. I'm Lenny Rome, the Vault Guy. Join me in my next video where I show you our first high-resolution structure of the vault. Do you think our prediction of the position of MVP was correct? Finally, if you find these videos useful or interesting, and you'd like to see more of the Vault story, subscribe and give us a like. I have no fantasies about these lessons growing so fast that they go viral. For now, I just hope we can grow slowly. Maybe we can go bacterial.